Firstly, the introduction of a 3D cell culture is a method to culture cells and grows it in all directions in the artificial environment to mimic how they grow in vivo, such as shape, cellular environment, and the ability to access nutrients, oxygen, metabolites, and signaling molecules. 3D cell culture also allows a cell to grow and interact with surroundings in all dimensions. What are the differences between 3D and 2D cell culture? The cell morphology of 3D cell culture forms in spheroid structure, less stiff, show drug resistant with the similar pattern that has been observed in patients. The culture can last from a few hours to a few days. Maintenance cost is more expensive, more time needed, and less commercially available. Then the history. In 1957, Aaron Moscona, an American development biologist, had shown that the cartridge cells that has been dissociated together create aggregate 3D structures. In early 70s, Robert Sutherland and his colleague tested the term as described by Moscona. They used a hamster lung cell and cultured it in suspension that create spheroids. In 1982, Mina Bezel, a scientist in the biological system and engineering division, showed the extracellular matrix that molecules outside the cells give structural and biochemical assistant impact in gene expression of cells than in 2D cell culture. Since then, 3D cell culture are widely used as it physiologically relevant.
Now, we will get into the procedures of 3D cell culture. There are a lot of different techniques available, so before choosing a technique, specifications like the natures of the cells must be considered to ensure the final goal of the study can be achieved. 3D cell culture techniques can be categorized as scaffold-based and scaffold-free. Under scaffold-based techniques, we have polymeric hard scaffolds and hydrogel scaffolds. And for scaffold-free techniques, hanging drop method, liquid overlay or low adhesion plates, and spinner culture are the mainly used techniques. In scaffold-based techniques, scaffolds function as physical supports where cells can aggregate, proliferate, and migrate in it. Their properties allow them to facilitate oxygen, nutriment, and waste transportation easily, and cells are embedded into the matrix, and the characteristics of the cells will be affected by the chemical and physical properties of the scaffold material used. Polymeric hard scaffolds provide 3D space to the cultured cells, so natural 3D tissue-like structures can be recreated. It can be made from different types of materials like ceramics, metals, glass, and polymers, and it can generate neatly arranged cells in vitro in a controllable and reproducible manner. For example, we have porous scaffold, which is one of the most used solid scaffolds, and this scaffold is significant for the tissue engineering applications like bone regrowth and organ vascularization. Next, hydrogel scaffolds. Hydrogels are used widely as scaffolds because they can resemble the extracellular matrix or ECM to some extent, while allowing soluble factors such as cytokines and growth factors to navigate through the tissue-like gel. They have soft and rubbery consistency, low surface tension, and high water content, which they are able to absorb a large amount of water, making them more preferred substitutes for natural tissues. Hydrogels are very practical for biomedical purposes, and they are also utilized in cancer research. Moving on, scaffold-free techniques aid the formation of multicellular aggregates known as spheroids, and various methods are available to develop them. The first one is hanging drop method. In this method, surface tension and gravitational force is applied to create droplets containing cells that in time will self-aggregate into spheroids with the help of gravity. This method has about 3 steps. Next, liquid overlay or low adhesion plates. This method applies materials that inhibit cell attachment to the surface of the plate, such as agarose gel or PHEMA that promotes the adhesion between cells, and cells will form spheroids spontaneously. This method is very simple and has only 2 steps. And last but not least, spinner culture. The use of convection force from the continuous stirring of the bar in the centrifugal flask bioreactor container helps in forming the spheroids. There are also only 2 steps in this method.
There are a lot of applications that are using 3D cell cultures. One of the applications is pharmacological applications and drug discovery. 3D cultures have the potential to significantly enhance cell-based drug screening and uncover harmful and ineffective compounds sooner in the drug development pipeline than animal or clinical trials. The 3D technique will be a simpler but more effective tool for drug genotoxicity, cytotoxicity, and the identification of anti-cancer drugs or agents. Drug testing in 2D delivers little information and is often deceptive, but 3D multicellular cell structures may bridge the gap between traditional in vitro and animal drug testing models. The manner in which cells are cultivated has a significant impact on how they behave pharmacologically. Changes in the cellular response to the medications, Paclitaxel, Ku174 and others were seen in a 3D model. The efficiency of 3D cell cultures in predicting drug-induced hepatotoxicity differed. Next, the applications for 3D cell culture is tumor models and application in cancer biology. Several characteristics of in vivo solid tumors have been shown by tumor spheroids in vitro. These similarities in treatment responsiveness between tumor spheroids and animal models might be attributed to their shared increased cellular connection through adhesion and production of soluble tumor factors that result in a low pH and hypoxia. Approaches from signaling pathways, expression, and interactions with ECM components, cell communications, variances, and cell proliferation rates and patterns are all examples of how 3D cell cultures may be valuable for cancer. And lastly, 3D cell cultures are used for cell proliferation and cell cycle research. Using three-dimensional cultures of mammary cells, vascular cells, mouse fibroblasts, and osteoblasts, researchers have gained a deeper knowledge of the processes involved in cell proliferation. Scaffolds with different elastic moduli were created by regulating the compliance of hydrogels using acrylamide. Cell proliferation was assessed in these matrices, and the data was utilized to investigate the mitogenesis control of the cell cycle in a tissue.
For limitations, let's see under cultures in concentrated medium or in gel-like substances. Firstly, the cultures in here is poor to reproduce between batches of biomimetic scaffolds, which can make it hard to extract all cells with increased size and tuberosity. Second, conditions of culture need to be taken with care, such as temperature and pH, and make it often difficult to use. Third, creating 3D matrices is difficult, time-consuming, and laborious, as it have too many components such as collagen chitosan, matrigel, fibrinogen matrices, alginate promatrix scaffolds, and polyethylene glycol matrices. Next is cultures on scaffold. The materials are required to construct the scaffold may affect the adhesion, growth, and cell behavior, and it also prompts the proteolytic degradation of single layers to split single cells from spheroid structures, which can take from a few hours to a few days. Then, scale of scaffolds and topography of cell distribution may cause various behavior of the cell. As you can see, this is a 3D structure stained with Epi. The blue one is the nuclei, the pink is the cells. Other than that, it is also difficult to do imaging depending on scaffold size, material transparency, and microscopy depth. And it is also need a greater flexibility to accommodate many cell lines. Under suspension cultures on non-adherent plates, the strong adhesion abilities of cells make some cell lines require expensive plates coated with specific materials such as polystyrene or covalently bound hydrogel.
Lastly, to summarize, 3D cell culture that was discovered and developed by biologists in 1957 to 1982 is simply a technique that allows the cell to grow limitlessly. This technology has various types of procedure to culture the cell in order to meet the specification on each technique to be successful. Some of the applications for the technology are available in pharmacological applications and drug discovery, tumor models, and application in cancer biology and also cell proliferation and cell cycle research. However, 3D cell culture may have limits to certain parts even though this technique has surpassed 2D cell culture, which has been used by thousands of laboratories for the last 4 decades. For instance, limitation included the state of the culture and also 3D matrices may be arduous to create since they have abundant components. Hence, it surely needs to be looked at thoroughly when using this technology so that it can give valid and precise outcome. Inclusively, 3D cell culture has already helped many tests and studies of the cells to thrive. Next is the evidence of the group work. We provide 3 different screenshots for the evidence. First picture is when we did the discussions for task division among group members through Google Meet. Second picture is when we discussed the journal that we wanted to refer before editing the video. And the last picture is when each member editing their part through Canva website. Last but not least is the evidence of the members' roles. These are some of the roles of each member during the group work of this assignment.